Brilliant. So why is UK Biobank so special? So there isn't any one factor that makes UK Biobank such a great resource that it is. It's actually a combination of several things. The first is that we all know it's pretty large. We've recruited 500,000 men and women aged 40 to 69, way back, feels like aeons ago now, 2006 to 2010, when they gave us consent to collect all of this data, recontact them, and follow up their health over time and make this data available for research purposes. The other aspect which makes UK Biobank particularly special is the depth of the data that we have. So we've collected an extensive amount of information on lifestyle factors, environmental factors, biomarkers, genomics, and so on, to enable researchers to look into the determinants of a wide range of diseases of middle and old age. The third factor is it's long. So because we recruited them 2006, 2010, we now have over 15 years of complete follow-up for all of these individuals, which of course means we now have thousands of instant cases of the most common diseases to enable really robust analysis to occur. And the fourth criterion, which arguably is actually been the most important factor as to why UK Biobank is being so successful today, is its easy accessibility to the data. So as you all are very well aware, the data from the very outset, the plan was to make all of these data available worldwide to researchers to perform research. So it's the combination of all of these factors. So when you compare UK Biobank to other major cohorts kind of within the same space, so here I've compared China Kadoorie Biobank, All of Us, which is recruiting 1 million participants across the US, and the Million Veteran Program, which is recruiting 1 million US vets into a research resource. You can see that we're all large, and actually UK Biobank is the smallest out of these particular cohort studies. But then when you start to think, okay, well, how extensive are the data that these studies have? You can see that actually UK Biobank has collected quite substantially more data than some of these other studies. We've also got sufficient duration of follow-up now, along with China Kadoorie, which also recruited about the same time as UKB. But All of Us and Million Veterans, they're still recruiting. So it's going to take 10, 15 years for the data they've collected on those individuals to be really relevant. And of course, accessibility. So UK Biobank is really the only longitudinal study that is really making its data easily accessible to researchers worldwide. So it's the combination of all of these factors that is what makes UK Biobank so particularly special. So just in terms of the access of the data, which is arguably, I think, the most important reason why UK Biobank has been so successful, we've got over 40,000 researchers who registered with us to use the data from academia and commercial groups, charity groups, from all around the world, over 100 countries now. The researchers are accessing the data, and over 10,000 publications up until quite recently. You can see that number is kind of increasing exponentially. It's about between 10 and 20 papers a day if you search on PubMed, are published on UK Biobank. So it's actually pretty tricky to keep track of it all. So in 2021, we launched our secure online research analysis platform. And this was primarily to enable researchers to access the whole exome and whole genome sequencing data, because there's just no way, with best will in the world, we could disseminate 20 petabytes of genomic sequencing data to researchers worldwide. So we launched the research analysis platform, which we call the RAP, in order to enable researchers to access the size and complexity of the data that they need to securely, to accommodate users with diverse skill sets and fields of expertise, and to enable scalable computing. So the RAP allows researchers to instantly access the data. There's no cost for you to store the data. You just have to pay for the amount of compute that you use. All of the data is in the same place. It's kind of in a research ready format. It's easy to explore with kind of the cohort browser that we have that sits on the front end of the wrap. Is a lower IT administrative burden because when you don't have to have large servers at your local institution to store the data, it's all done on the cloud, easy access to tooling, and as I said, large scale and specialized compute. So we now have about 7,000 researchers who are actively using the wrap at the moment, and we plan to, hopefully that number will increase quite rapidly over the next few years as more, it's a bit of a steep learning curve to use the wrap, but once you kind of got your head around it, researchers I've spoken to said basically they don't look back. It's absolutely fantastic. You can do analyses on the fly really quickly. Everything's there at your fingertips. So you can see here, this is the number of research projects that have been undertaken using UK Biobank over time. And that the number is just, obviously, as you can see, it's increasing from academia, but also in particular from commercial companies are now actively using the data in anger. So over 90% of the top 200 universities in the world are using UKB data, and over 70% of the largest pharma and life sciences companies are now using UK Biobank data. So, and I just want to segue into just to showcase just some of the scientific discoveries that are being made by UK Biobank. And first of all, I just want to kind of take us in chronological order and focus a bit on the research that's coming out from the genomic data, then the imaging data, and then the accelerometer data. So, as most of you are probably aware, in 2017, we released genotyping data for all 500,000 individuals. And I don't think it's an underestimate to say that was a bit of a game changer, actually, both for us and for the research community, because for the first time, it enabled researchers to do large-scale GWAS analyses on a single data set rather than having to pool multiple smaller case control data sets. So that led to an explosion in the literature of GWAS, and also led to quite a few online browsers that showed summary statistics from GWAS of all of the phenotypic traits that exist in UK Biobank, of which there are now over 10,000. So if you look online, there's GWAS browsers for phenotypes, both from Stanford, from the Broad, and from Edinburgh. And so this just demonstrates just results taken from the Gene Atlas browser. So you can see GWAS for a continuous trait like standing height, you can see that actually thousands of hits that are related with standing height. Whereas if you look at a binary trait here, celiac disease, you can see that the association is somewhat quite different. It's actually only relatively few SNPs that are strongly associated with this disease. So this is the sort of information that you can obtain from these GWAS browsers. And that information allows you to do Mendelian randomization studies. I don't need to tell this audience about MR, but it's really helped to move the field forward so much quickly in terms of us trying to understand the potential causes of disease. Because you can take these SNPs identified from the GWAS, use those as proxies for exposures. And these MR methods are evolving really rapidly, particularly over the last couple of years. And there are now online platforms. I'm just showing MR-Base as an example, produced from the group in Bristol, where it really facilitates researchers to use MR methods based on previous GWAS to look at causal inference in relation to a whole range of phenotypes. So the work from these data from UK Biobank has really helped to accelerate that field really quickly. And of course, it's also led, the GWAS data has also led to the development of polygenic risk scores, or PRS scores, that I'm sure all of you are also really well versed in. I don't need to tell you about these, but it's really been the UK Biobank data that's led to an explosion of developing PRS scores and then thinking about how can you apply polygenic risk scores together with all of the lifestyle information that exists in order to create a prediction score for individuals as to the absolute risk of developing disease. So what you can see here, this is taken from data from Genomics PLC, that for many chronic conditions, individuals in the top, say, 3% of polygenic risk of developing a condition actually have between a 20 and 30% lifetime risk of developing the disease, which is pretty similar to many monogenic disorders. And so you can see how if this type of genetic profiling, if you like, was introduced into clinical practice, how that could be used to then potentially identify individuals who would benefit from, say, earlier or more intensive screening for breast cancer, or being put on preventative statins because they have a high genetic risk of developing cardiovascular disease, as an example. So this has only been possible because of the large size of UK Biobank, the standardized assessment of all of the variables in the resource, and the long scale of health follow-up that we have. So I also wanted to showcase other research that's come out of the genomic data that could be used to help clinical diagnostics. So this is taken from a paper from the Exeter group a couple of years ago now, and what it shows is that the polygenic risk scores for people with type 1 diabetes are actually quite different to people with type 2 diabetes that you can see here. And what they showed was that actually about 40% of the individuals in UK Biobank who had the polygenic risk score for type 1 diabetes were actually misdiagnosed as having type 2 diabetes in middle to old age. So that has obviously led to incorrect diagnosis and obviously incorrect medication. So what that's led to is clinicians being more aware that if they have patients who they've diagnosed as type 2 diabetes in middle age and they're not responding to usual medication, that perhaps they ought to consider maybe they have type 1 diabetes instead. So these type of data already starting to infiltrate into GP practices in terms of trying to better differentiate clinical diagnostics, particularly for disease subtypes. So on the back of the genotyping data, we then went on to perform whole exome sequencing. And this was done through an investment for pre-competitive consortium funding through a group of largely pharma companies. The first set of data was released a few years ago. The data for all 500,000 was released in 2022. And the real value of those data are now really starting to come out in research papers that you can see pretty much every week from UK Biobank. So this example just highlights the value of these data. So if you see that what I've circled here on the bottom, this is the relationship between variants in all of these genes in relation to body size. And what you can see, there's a rare genetic variant in this gene at the bottom here, GPR75, that is related to individuals with a low body weight. And that has then led to pharma companies considering, well, could we use this as a potential therapeutic target? Could we create a drug that mimics the function of this loss of function variant in order to induce weight loss? Then on the other end of the spectrum, you have another rare variant in the gene called BSN, lovingly known as the bassoon gene, which I particularly really like. And this rare variant in this gene is related to a large increase in body size. And interestingly enough, this bassoon gene, we know, is involved in the release of neurotransmitters in the brain. So it's starting to lead to researchers to think about novel mechanisms through which adult onset obesity develops. And it's starting to lead to us to think, well, maybe some types of obesity might be related to neurodegeneration because variants in this gene has such a large effect, particularly on adult onset obesity. So the whole exome sequencing is starting to lead to accelerated drug discovery and also starting to lead to a better understanding about the mechanisms through which some diseases can develop. Then quickly on the heels of whole exome sequencing, we started to release data on whole genome sequencing. And this was enabled by a consortium of charity, government funding and industry that came together. And the release of the whole genome sequencing data on all 500,000 was released the end of last year. And about 95% of the population, you're probably all well aware of this, of white ancestry. But actually, there are about just shy of 10,000 UK Biobank participants with whole genome sequencing from South Asian ancestry and from African ancestry. Now, it might not be particularly large numbers in our field, but nonetheless, at the moment, it's still the largest data set in the world that has whole genome sequencing on these types of ancestries. And the beauty of having WGS data on 500,000 individuals is it means as sample size increases, you can see here the number of rare variants identified also increases substantially. So what's the value of having whole genome sequencing if we've already got whole exome sequencing? So, I mean, the obvious answer is you collect a shed load more data on the variants because you're covering variation across the whole genome. So you've got data on about 1 billion SNPs and about 1 billion indels. And it also captures variation across the 5 and 3 untranslated regions of the gene, which we know are important for the development of some diseases. And interestingly, it also captures a bit more of the genetic variation within the exome compared with whole exome sequencing, just because it's more detailed sequencing. So whole genome sequencing data will for sure enable novel research into how genetic variation, both across the genome and the exome and the non-coding regions of the gene, influence disease biology and disease risk. And it's just worth emphasizing that a resource like this with such granular data, coupled with all of the other data we have on lifestyle, environment, biomarkers, health outcomes, just doesn't exist anywhere else in the world today. So moving on to proteomics. So a few years ago, a consortium of quite a few pharma companies that you can see here came together to invest into UK Biobank to perform an initial study of measuring 3,000 circulating proteins from the Olink platform in a subsample of just over 5,000 UK Biobank participants. Those data were released, the final data were released last year, and you can see this was the first paper that came out actually that looked at genetic associations between these relative abundance protein levels. And what they found was that there's actually over 10,000 genetic associations identified between genetic variation and protein levels, the vast majority of which were actually newly discovered. And this has led to great excitement as to, well, you know, if we've got these data on 50,000 participants, what will be the value of having these data on the full cohort if we were to be able to scale this up? So early analysis so far from these companies that have been published in the literature are focusing on the value of proteomics to produce early detection biomarkers and as diagnostic markers with incident disease. So, for example, this is a paper published late last year by a group in China that used the proteomics data to show that actually there's a relatively small number of proteins that are strongly associated with the subsequent risk of different types of dementia within, you know, over a 10-year period. So it starts to lead to the research field to think, well, you know, can we actually develop a blood-based marker for future risk of dementias based on something like a proteomic panel? It's also led to research on disease biomarkers. So can we create a diagnostic biomarker for disease based on proteomics? So you can't really see this very well, but what it essentially shows is that proteins are differentially either upregulated or downregulated in a whole range of different inflammatory conditions, whether you've got MS, celiac disease, Crohn's disease, ulcerative colitis. So you can start to see how you can use these protein panels potentially to help in the diagnostic pathway, particularly of some of these types of inflammatory conditions. So hugely exciting area in the role of proteins in disease prediction, but also disease diagnosis. So then moving on to what's the value of having wearables data. So by wearables specifically here, I mean the wrist-based activity monitor that we gave to 100,000 participants. It was 10 years ago now, actually, to wear for a period of 7 days. And those data are really starting to show some really exciting results. The first is pretty obvious, actually, but it's really nice to see it in black and white. We all know that self-report of physical activity is pretty crap, but what you can see here is you can really see that in black and white. So when you look at the relationship between physical activity and all-cause mortality, the relationship is much stronger when you assess physical activity using an objective marker based on a physical activity monitor versus self-report. So the physical activity data has also been shown, this was research again published late last year, to be able to predict Parkinson's disease up to 7 years before clinical diagnosis, which is pretty astonishing actually if you think about how many people are out there walking around with some kind of wearable device, that it could actually predict your risk of developing Parkinson's several years in advance. And it was basically simply on doing lower levels of physical activity. So the bottom 2 lines on that graph are those participants who either got prodromal disease or have a diagnosis of Parkinson's. You can see their profile from the physical activity monitor is much different compared with people without the disease. So moving on to imaging, as most of you are aware, we're nearly at the end, actually, of performing an initial imaging scan on up to 100,000 individuals. This is by far the largest imaging study ever conducted. Consists of a brain MRI scan, a heart scan, what we call a body scan, which is actually neck to the knees, a full body DEXA scan to measure body composition and bone mineral density, carotid ultrasound, and a 12-lead ECG. It's a pretty intensive visit, 4.5 to 5 hours. The participants literally spend all day with us, and some of them travel up to 100 miles to come to one of these assessment centres. So it's a huge ask of participants to do this. We've already scanned over 80,000 of them, and we plan, hopefully we'll get up to 100,000 by the end of this year. And then over the next 6 or 7 years, we plan to bring back 60,000 of those 100,000 participants back for a second scan in order to enable researchers to look at change in internal physiology over time and how that relates to subsequent disease. And in addition, for those participants who are over 65, we also give them the option of going home with a cardiac monitor that kind of sits on their sternum that assesses subclinical atrial fibrillation. But as an added bonus, it also gives us data on physical activity, sleep, and gait measures as well. So Aidan in our department is currently looking at those data as we speak. So, you know, what really is the value of having multimodal MRI scans in a large-scale resource like UK Biobank? So the first is biomarkers. So I don't mean blood-based biomarkers here, I mean imaging-based digital biomarkers. So this is an example published last year where researchers looked at the heart around the fat around the heart that could be used as a pretty good biomarker for heart failure in those already diagnosed with the condition, but it could also be used to predict those who would go on to develop heart failure within about a 5-year time frame. So, you know, it's these type of research just hasn't been able to be done before. So pretty exciting. And the second thing that the imaging data is really useful for is to help us to understand mechanisms of disease by ascertaining intermediate phenotypes. So this research is looking at the relationship between various measures of body size and the brain age gap. So the brain age gap, what I mean is individuals who have an older brain based on various neuroimaging markers, particularly white matter hyperintensities, compared to their chronological age. And what you can see here, the red blobs are men, the pale blue blobs are women. What you can see is that the relationship between various metrics of body size and accelerated brain aging is generally stronger in men than women. But what caught my eye and I thought was particularly interesting was if you look at the percentage of body fat, you can see that the relationship is almost diametrically opposed. And this is interesting because it kind of supports underlying hypotheses that postmenopausal women who have a high percentage of body fat have generally higher circulations of estrogen that comes from the adipose tissue that is beneficial for brain health. So you can see that relationship there really clearly, and it's very, very different to men. So it starts to, you can start to think how you can use the imaging data, not just as biomarkers, but also to really start to think about the mechanisms through which, for example, body size may influence brain health. Another interesting example, again, published a couple of years ago about the value of imaging is kind of an unintended consequence of the imaging, if you like. We implemented the DEXA scan to enable researchers to look at body composition and things like osteoporosis and bone mineral density. What researchers here from Southampton have been able to do is to show that you can use these scans to clinically identify hip osteoarthritis. Up until now, hip osteoarthritis is currently, the diagnosis is quite subjective based on symptoms and visual examination of x-rays. But using the DEXA images from UK Biobank, researchers have developed kind of machine learning algorithms to identify hip osteoarthritis, which is correlated with clinical metrics like hip pain, for example, with huge implications for how that could be rolled out into clinical care. So pretty exciting. So the final bit, I just want to kind of move on into plans for the future, certainly for the next sort of 5 years or so, and what we'd like to do with a resource. So I think we can group this into kind of two main areas. One is how we can enhance the characterization of risk factors in our participants. So this would be through extended assays conducted on our stored biological samples and by performing a repeat assessment on all surviving participants to collect additional metrics on risk factors. And the other area is what can we do to enhance the characterization of the health outcomes that our participants are now experiencing through extended linkages, specific assessments of people with certain conditions. Could we develop an app that could track people's health over time, for example? So for the cohort-wide repeat assessment, what we'd really like to do is to bring back as many surviving participants as possible to repeat what they had at baseline and also to add some new assessments. So the value of doing this is that by repeating the assessments that they had done 20 years ago, it will enable researchers to look at how change in lifestyle, change in environment, and perhaps more importantly, change in circulating biomarkers over that time period, how they related to subsequent development of disease. Because looking at a change in a risk factor is statistically much more robust than looking at the association with a single point of time. Similarly, it will enable researchers to look at how change in functional health outcomes, whether that's cognitive function or change in blood pressure or what have you, how are they more strongly associated with previous exposures taken at baseline? Of course, while we've got the participants with us in the assessment centre, it also provides us with opportunities to collect new information on them. So, for example, given the average age now is 70-71, we're particularly interested in collecting new measures related to aging and frailty. So can we collect other measures on cognition, memory, speech, that is all really relevant for prodromal stages of neurodegenerative disease? Can we collect measures on strength, balance, movement, gait through wearables, getting them to do various tests in the clinic or online questionnaires? So these are all the things that we're starting to scope out now. So in terms of expanding the linkages, you can see here that over the next 10 years, there's going to be an explosion of health outcomes in UK Biobank participants, with tens of thousands of incident cases of the most common conditions. So what we would like to do is to link to, in addition to the HES data, the hospital admission data, the cancer, the death, and fingers crossed, primary care data, we're also actively pursuing whether we can link to these other types of clinical audits and other health-related registries to really add value to the characterization of health outcomes. And at the moment, we're implementing new software tools here in NDPH so that we can streamline how we process, curate these new data sets to make them more rapidly available to the research community. So the other thing we'd like to focus on is, can we characterize cancer subtypes better? So we have information from the cancer registry, but what we'd really love to be able to do is to be able to link to digital histopathology images to enable researchers to identify molecular subtypes of cancer. So to look at the genetic and environmental determinants of very many different types of molecular subtypes. So we plan to do some pilot work, some of which is with a company called In Sitro in the US, to assess the feasibility of being able to link to these images, run algorithms on these images to develop these metrics based on the imaging scan that can sub-characterize cancers into different types of phenotypes. We're also about to conduct a pilot study with Genomics England to assess whether we can link to their somatic tumor mutational data that they hold on participants who are parts of 100,000 Genome Project, who are also parts of UK Biobank. And again, similarly, to enable researchers to look at the somatic germline mutations together with the host genotype. We'd also really like to focus on can we characterize neurodegenerative disease in much more detail than we currently can do. So we're currently scoping out a pilot study called a brain health assessment to see if we can subtype people with different types of dementias. So this will involve participants having a brief MRI scan to assess brain pathology, to collect a biological sample to measure neurodegenerative and other omic biomarkers, and also to collect a symptom or cognitive function survey to assess the clinical presentation. So what this type of enhancement will enable researchers to do is to be able to accurately subtype whether a participant has Alzheimer's, vascular dementia, Lewy bodies, Parkinson's, what have you. And there may be other types of subtypes that we just don't know about based on the data that we've collected to enable research into their potential causes. And it also provides us an opportunity to give participants wearables to assess gait and speech and so on. So finally, we'd also like to expand the sample assays. We know that they provide a huge wealth of information to researchers, but in order to do this, it's become pretty clear to us for some time that we need a new sample archive. At the moment, if you want to pull out all 500,000 samples taken at baseline, it'll take us about 2 years, just because the freezer systems and the storage systems we have up in our sites up in Stockport are 20 odd years old, and they were not designed to retrieve cohort-wide samples. So we need a new sample archive where we can pull multiple samples out in a parallel way, and hopefully will speed up the process by at least fourfold. And this new sample archive obviously needs to be adjacent to labs, the staff, to scientific input. So we've managed to secure funding a couple of years ago, whereby we can procure new sample archives, new freezers, new LIMS systems, and a new building in the University of Manchester, rather than the current offices in Stockport, to really facilitate some of this work, with the proposed move being about in mid-2026. So we're very optimistic that once this new sample archive is in place, that will be able to support cohort-wide assays of a whole different range of different assays that are currently in the mix. So areas of interest, some of which are more mature than others, includes cohort-wide markers of the immune system, doing cohort-wide proteomics, metabolomics, using mass spec rather than NMR approaches, infectious disease biomarkers, transcriptomics, and epigenetics as well. As I said, all of these are in various stages of excitement and progression, but certainly over the next sort of 5 years, I would hope that we will be able to progress all of these to a greater or lesser extent. So I just want to leave you with this final whizzy slide to inspire you to think about, actually, are there any other things we could be doing that would help you in your research? So, you know, if you have got any ideas for, I'd really love to send out a web-based questionnaire to participants to ask about, you know, X, Y, or Z. I really think you should be measuring this type of device that would really help move research forward in this field. Please do come and have a chat with us, because now is the time to do that, because we're starting to scope out what some of these new enhancements look like. And just before I end, finally, just a heads up that we're planning to introduce a series of seminar series in September for kind of early career researchers across the campus who are using UK Biobank data to kind of start a discussion and about what we're all doing. I have no idea. I know what some of you are doing, but generally speaking, I don't have a good feel for all the fantastic research that's been done, not just in NDPH, but across the campus. So it would be great to get your early career researchers to come along, tell us about the research you're doing, you know, to collaborate more, and to also tell us at UK Biobank about how we can make the resource better for you. So